We work in a disease called chronic lymphocytic leukemia, which is a mature B cell disease and mostly identified by aberrant expression of CD5. And about 10% of CLL patients progress to a more aggressive form of a disease, which is mostly lymphoma, which is called as Richter's transformation. So the current challenges in Richter's transformation is there are limited models and the patients do not survive more than a year. And there is an unmet need to understand the molecular mechanisms. So in Richter's transformation, one of the major alterations that is there is the MYC alterations, which is observed in like 60 to 70% cases. And recently, from the whole genome sequencing analysis from Nadu et al. And from Perry and colleagues, recently, what has been identified is Richter's can be classified into subtypes and almost like 5 subtypes. And what has been observed is there are mutations in negative regulators of MYC, especially a gene called MGA, which is MAX gene associated. This gene is a functional repressor of MYC, and it is often lost and undergoes loss of function mutations, acts as a tumor suppressor. And it's observed about 30% of cases can have MGA mutations in Richter's. So, and previously in CLL, 13q deletions and mutations in splicing factor SF3B1 have been known as the most commonly occurring genomic alterations. And given these observations, the genomic observations, we wanted to utilize this to develop a mouse model. So we developed a B-cell restricted model where we have 13q deletion, MDR, which is mimics 13q deletion, and SF3B mutation. And we perform MGA deletion using CRISPR-Cas9 in early progenitors, which is LSK cells, and we engraft them in the immunocompetent mice and follow them by serial bleeding for 24 months. And what happens is basically what the B cells develop after a few months, and we follow the development of the disease, and we observe that 3 out of 15 mice develop CLL, which is an aberrant expression of CD5 and BT20 in the mice. And when we took this splenic B cells and put it in immunocompetent or compromised mice, we saw the loss of CD5 expression, and there was an aberrant expansion of the B cells. Now, when we further examined these B cells, we saw that it was increased Ki67 and increased MYC expression, and with features such as the large size, and it was mimicking the human Richter's transformation. And now we know that we know that human Richter's basically has a large size and increased Ki67. So we serially transplanted these cells, and we observed that the development of the disease was similar. So we further examined this Richter's and CLL, and we saw that there was an increased OXPHOS signature and increased MYC signature from our transcription. And interestingly, we observed from our electron microscopy analysis that the mitochondria from these Richter cells are very abnormal from CLL and normal B cells. So our findings indicated that this dysregulated mitochondria may produce aberrant changes and functional changes in the mitochondria, leading to increased cellular loss, which are basically the byproducts of OXPHOS. And this can trigger oncogenic signaling. And further, we observed that this Richter mouse cells were utilizing TCA cycle substrates more than other metabolic substrates, indicating that OXPHOS is one of the major players here. And from our functional cell line-based experiments, we observed that one of our targets, which is basically called NME1, was upregulated, which is a nucleoside diphosphate kinase. The same target was also observed in our Richter cell. We further validated this in human Richter samples as well, indicating that NME1 could be a potential target. However, NME1 doesn't have any commercial inhibitors. So we had to target this gene via indirectly. So we figured out that MYC regulates NME1 and MGA also regulates NME1. The difference is that MYC is a positive regulator and MGA is a negative regulator of NME1. So we validated this using cell lines, and we further wanted to target this. So we used a CDK9 inhibitor, which is basically targets MYC, called as AZ5576. And since we observed OXPHOS and we found that mitochondrial OXPHOS complex 2 was upregulated in all our samples, we utilized an inhibitor of complex 2, which is called as TTFA, tetraphenyl fluoroacetone. Using a combination of complex 2 OXPHOS inhibitor and CDK9 inhibitor AZ5576, we found that the Richter mice can live significantly longer than the single treatment controls, indicating that this combination of OXPHOS inhibitors and CDK9 inhibitors definitely improve the survival, at least in the preclinical models, indicating that this could be a potential new target in the field of Richter's transformation. Targeting this axis, this MGA-MYC NME1 axis, provides a new potential target, and these findings open up new avenues, developing targeted therapies to treat Richter's transformation more effectively.